June 2006. Hans Clevers still remembers the day clearly. For more than 5 years, the Dutch molecular geneticist had been doing research into the human intestine and its amazing ability to regenerate itself. Every 2 to 4 days, the intestine renews its entire surface. No other human organ regenerates faster. The researcher had long suspected that special cells, hidden somewhere in the intestinal tissue, were the ones responsible for this constant renewal, the so-called adult stem cells. Fortunately, there are stem cells in every organ. They can divide and replace cells that are lost. Usually, adult stem cells are not very active, but as soon as damage occurs in an organ, they begin their work and replace the lost cells. But nobody knew exactly where these special intestinal cells were located. They were thought to be small and rare, and therefore difficult to find. But Clevers and his team had an idea. The surface of the small intestine consists of countless villi, which the body uses to absorb nutrients. Between the villi, there are small depressions, so-called crypts. This is where the researchers suspected that the adult stem cells were hiding. To find them, you have to have a special tool. We found a molecule called LGR5 and realized that it was located at a cell that was a promising candidate. The researchers believed that the stem cells they were looking for were different from normal intestinal cells because of a molecule called LGR5. Therefore, using a complicated technique, they colored all the small intestinal cells containing this protein green. Since the method works on living organisms, they could now observe how the cells behaved. The result still delights Clevers even today. Nick Barker and I were looking through the microscope at the same time, and we saw some very small cells glowing green. We'd spent 5 years trying to figure it out, and now we both knew right away that we were looking straight at the stem cells. The researchers were able to watch the stem cells at the bottom of the crypts dividing and then migrating up to the tops of the villi. The discovery was a scientific sensation, but it still didn't fully satisfy the researchers. We noticed that the stem cells in the intestine weren't behaving in the way you'd expect them to. They were dividing each day. We realized that if they could do that in a body, in tissue, then surely they could do it in the lab as well. And the stem cells began to grow in the lab indeed. Millimeter-sized cell clumps formed. With the aid of the LGR5 molecule, the researchers isolated stem cells from the liver and other organs. Here too, they successfully cultivated artificial mini-organs, so-called organoids. We can now take a small amount of tissue from any organ and any individual and grow it in the laboratory. Patients could soon benefit from this discovery. 15-year-old Dunia, for instance, who suffers from the genetic disorder known as cystic fibrosis. Because secretion in the lungs and other organs malfunctions, patients have problems with digestion and breathing. Without therapy, the disease is fatal. The normal level is marked in green, and here you can see how much air Dunia managed to blow out. At her age, lung capacity is normally around 4 liters, but Dunia has only 2.5. Antibiotics and many other medicines have so far only managed to relieve the symptoms. New drugs are fighting the disease at its roots. Since it occurs in many subtypes, there are also different types of these new drugs, and that's a problem. These are fairly expensive drugs. They cost $500,000 a year. With our very cheap and fast test, we can measure whether the drug is really having an effect on the patient. If our test is positive, the patient responds. If it's negative, we don't administer the drug at all. Using organoids grown from the patient's tissue, tests are carried out to see if a particular type of drug works, because every person reacts differently. Tests on sick patients are no longer necessary, a big step towards personalized medicine. Organoids are also playing an increasingly important role in the development of new drugs. In a biobank, Clevers collects tissue samples from patients, and the researchers then test the new drugs on organoids grown from them. Ethically controversial animal experiments can therefore be replaced more and more often. And Clevers has big plans for the future.
Mijn droom zou zijn dat we ergens zoals een bloedbank... dat
My vision is that just like a blood bank, we have an organoid bank. And if someone needs a new liver, for instance, we can look in the organoid bank and say, OK, this tube is best for your liver. We then thaw it out and administer it to the patient. He goes back home and the stem cells start regenerating his liver. Because stem cells are very good at doing just that. Clevers is hoping for organoids to become normal in clinical practice in just 5 years. For the researcher, it would be a further milestone in a life rich in discoveries.